After the temperature and the pH, what are the other environmental factors which can affect the virus life, or which are capable of killing the virus? Most precisely, we can see it. Maybe it is the drying temperatures, or maybe it is the dry the virus, or maybe it is the shaking of the viruses. So we will see it, the drying and the shaking of the viruses, and how much it can affect the virus life. Most viruses withstand with the freezing temperatures, with the drying, with the frozen state. Because freezing is the most common procedure, we can store the viruses. This is the reason that we store the viruses in the frozen form, because most of the viruses, they can withstand with the freezing. And freezing, maybe they are not capable of course the viruses are not capable of reproducing without the any host cells or without any host. So of course it is very common to save them in the frozen form. Identification brought by the oxidation during the drying process. During the drying process, with the help of the oxidation, maybe inactivation is very common procedures. Presence of the foreign proteins in the medium experts protection may be also one of the reasons for the drying with the viruses. We can see it here, the lyophilization of the virus. After the lyophilization, what kind of results? We taken the example here, and we see it TDCID, the tissue culture infectivity dose 50 of the lyophilized virus stored at the room temperature and at the 4 degrees centigrade. And they were capable of reviving for about 1 month, 4 months and 6 months. You can see it here. We have taken the example of the adenovirus, and here is a research was done, and we can see the infectivity of lyophilized adenovirus and different time intervals. The used material was the tissue culture ID 50, and the time of storage after lyophilization. They are quite capable of stored at the 1 month, it's enough, and then 4 months, still 6 months at the room temperature and at the 4 degrees centigrade. The value of the TCID 50 on the y-axis correspond to the 10 years of different samples of the adenoviruses. We have taken the different samples, and we have taken it for the 10 years to see its effect. Here is one more example by the researchers we have taken for the lyophilizations, and the virus was revived at different time intervals. The value of TCID 50 on y-axis was corresponding again to 10 years of different samples, and the samples were again we have taken as the adenovirus. It was from the same results, and you can see it here that the stock of the virus, and then 1 month they are quite capable, and 4 to 6 months similar conditions were taken for this experiments. It is the same experiments, a different way. Viral load of the lyophilized virus stored at room temperature and 4 degree for about 1 to 6 months. Similar experiment. Here we can see the virus load, which we have calculated in the 100 nanogram of the DNA per 100 nanogram of DNA, and this is the viral load, and this is the 1, 4 and 6 months. Still we can see at the 6 month viruses are quite capable of living for the lyophilizations. Why this protection has been given by the drying or by the freezing temperatures with the, it is because of the proteins which are present inside some specific kind of the virus. When viral suspension is shaken, the similar kind of the protection can be provided to it, and it may be not so easy. However, it is very easily the virus can be inactivated with the shaking. The capsid of virus is something like the shell of the turtle, which is very strong and which is capable to make it virus not easy to be deteriorated with structure. If the shell can be compromised by the mechanical variations, the virus will be inactivated. So it is very important when we are doing in different kind of the experiments in the laboratory, that viral spec will be done very, very carefully. We have already seen in the laboratory techniques that virus is very shaken. With the shaking, the virus will be inactivated. So it is very carefully should be taken, even especially when we are doing the different laboratory experiments for with the viruses. So we should be very careful with the viruses. Even we are working with the test tubes, we need to be avoided the external shakings or physical methods. And similarly, even in the test tubes, it is very easy that virus will be shaken. So the care should be taken when we are working with these viruses. All objects resonant frequencies at which they naturally oscillate, of course, because with the drawing, pluck the guitar string and it will be vibrate at the resonant frequency. These frequencies are similar with the viruses, but resonating can get out of the control. Of course, a famous example is the Tacoma Narrow Bridge, which wrapped and finally collapsed at 1940 due to the wind that rocked and bridge back and forth at one of its resonant frequencies. Whether the similar position will be given to the viruses or not, viruses are susceptible to same kind of the mechanical excitation. So with the experiments which were designed by the Katie Sun from the Arizona State University have shown us the same resonance can be shown by the viruses by the pulses of the laser light and can induce destructive vibration in the virus shells, just the similar way their resonance which we have seen in that bridge. So how to calculate this resonance, whether it will be enough for the virus to kill it or not? It is very difficult to calculate what sort of the push will be capable to kill a virus. Since there are millions of the atoms in the shell structure, of course the capsid of the virus is very important and they have the million of the they can be direct computation of the each atom's movement would take the several 100,000 gigabytes of the computer memory. It is unlikely that virus will develop a resistance. However, if we are capable to produce some kind of the mechanical things to inactivate the viruses, it will be a very important discovery. If we see the mechanism how to inactivate with some specific kind of the push to inactivate the virus, it will be very important discovery and it will be very difficult to develop the resistance to mechanical shaking type of the treatment of the viruses as do with other drugs which we make them the resistance, and that's why it will be difficult to use the same drug again for the same kind of the disease.